As Pradeep introduced me, I am a scientist, a cognitive neuroscientist from National Brain Research Center, and I've been working there for the last 7 years. And my primary interest is in human perception. So what we have worked on till now is some studies we have done on multisensory perception with variety of techniques. We have studied humans primarily using electroencephalogram, which is EEG, which some of you might have heard about. But more generally speaking, it is very much a technique similar to ECG, which is done on heart. But in the difference being that it is now being done on brain to understand the brain signals. There is also another technique which we use called the fMRI, which is functional magnetic resonance imaging, where you probably have heard that you, or probably some of you had been to an MRI scan where you can see a bunch of structures. Either it is from the brain or it can be from the different parts of your body, such as the bones. But what these scans also do is give us very precise measurements of how the brain functions in some order of some seconds, right? Now, all the work that we have done till now is using these different kinds of data sets. And these data sets actually target different levels of nervous system organization, which I will talk about briefly. But what is interesting that is happening with this is that we get large volumes of data. And what you also right now get is this different big projects that are coming up. The Human Brain Project from the European Union, the Human Connectome Project, which has involved scientists from US and also Europe. And we have also something similar, like China has one Human Brain Project as we speak. And similarly, India has a couple of things on the cards. And one of the things that we are also interested in currently is a program on mental health, where we are in the long term, in the next 10 years, we are going to record data, multimodal brain data. So to cut the long story short, so we have, we are going to get enormous amounts of data. And now I just gave you some hint of the data that we are collecting at the level of humans, but we are also going to collect, and some of us have already collected this data at different organisms, such as C. elegans, which is a worm. There is the mouse, there is the monkeys. So we can collect all this data from different species, and that gives us also very important understanding about brain functions. Particularly, there are certain things you cannot do with the humans, some kind of experiments, and those we can do with the monkeys to come up with new drugs, new sort of therapeutics, brain-machine interfaces, something where we need also a lot of engineering people. So, however, I mean, I am not going to talk about, as you realize, about all of these. It's practically impossible. But my talk is going to be something more fun today. And more importantly, what I would like to have is to ask questions, or rather, I hope that you go out with this talk with more questions rather than answers. Yeah, so let me try to share my screen now. And please, Pradeep and the team who have, by the way, organized a fantastic meeting, I guess. So which I was enjoying the earlier videos also when I was seeing through them, like how to prepare my talk. So if anything goes wrong, please help me, okay? Okay, there we go. So I hope everyone can see my screen. Let me sort of go back to the main thing. Okay, so as I said, the title of my title of the talk is a bit provocative, and necessarily so, because I think grand unified theories of brain function is something very much fun to discuss about when we are sitting at home mostly nowadays. And so this is a Saturday. And my talk is going to be divided into two parts. First part, I'm going to talk about the grand unified theories of brain function, which scientists have been worked till date. And then I'm going to talk about the insights that we have learned from my studies, where several people are also involved, and the insights we get from the studies of multisensory perception. So first, to start off with the grand unified theory of brain function. So I would like to show you the pictures of these two scientists, because the history is important, and what they kind of came up with this neural doctrine. One was Camillo Golgi. And so some of you probably in high school biology encountered Golgi bodies. You are very much right. This guy, this Golgi bodies in the cells owes their name to this guy, Camillo Golgi. What he came up with is a unique way to stain different cells and see them under the microscope. And what he observed are these following structures, where we saw the brain cells in a tissue can be structured like these wires, which we now know as the neurons. These are actually individual brain cells. And so what Golgi proposed, though, that these neurons are some sort of a connected kind of, you can think of like some kind of a bed sheet. So which the sheet is connected by each different kind of thread. So these are these threads, but essentially the brain is a continuous matter like this. And this theory, what he called was this reticular theory of brain function, where essentially our thoughts, our actions, everything are governed by the activities this continuous sheet will have. So this is what Golgi sort of talked about in terms of the grand unified theory. Then Santiago Ramon y Cajal, he was from Spain, and what he was doing his experiments, I think, in Madrid, and what he kind of used the same techniques as Golgi invented, and he was very much, inspired by them. But what he came up with this idea, maybe they are not continuous sheets, but what he came up with this idea is that these are actually individual cells. In other words, they are individual functional units of the nervous system, meaning these cells themselves are computing in terms of I am assuming I can't see the speakers here today, but I am assuming a lot of you are engineers, like the organizers of this team are. So these cells are something like information processing devices, which are computing actually. And this word is very important here, that these cells are actually computing information in the brain, right? So this is the concept of this information being computed in the nerve cells. You can owe their allegiance to, the idea can be owed to Santiago Ramon y Cajal. So in the meantime, when this was going on, these developments, there was this idea, there is this neurologist or rather neurosurgeons, because in those days, neurosurgeons and neurologists were kind of similar people, but nowadays they have become different disciplines. But nonetheless, so there were different people like Paul Broca, Brodmann, and Wilder Penfield. They were looking at sort of these surgeries, people who had surgeries such as these lobotomies, where they can study the brain function in detail. And when they kind of, for example, Paul Broca, when he targeted a specific area in the brain, and I hope you can see my cursor here. So this area, if this area is sort of activated so it is stimulated or it is resected, people lose their ability to speech. Right, so this area was related to this guy is some kind of a speech area. So similarly, different kinds of brain kind of areas can be attributed to different kind of functions, such as, So in the back of the brain, you have the visual processing area. Here you have the auditory areas. You have the planning and organization areas here. Speech comprehension areas are here, which is connected with our hearing. So all these idea is that all these areas are kind of segregated in the brain, which are kind of modules which are processing some kind of information. Now, there was also this guy, Charles Arthur Sherrington, who actually talked about how these nerves, which are the cells that Santiago Ramon y Cajal talked about, are connected now, and the connections can be both excitatory and can be inhibitory, and the point at which there is a connection, he termed them the synapse. The synapses are very important for propagation of these signals, and if we understand the activity or these excitation inhibition profiles of these cells, or in the cells, the electrical activity, we can understand a lot about this computation. So in a way, he talked about how the information might be integrated over the group of assembly of cells. So imagine your hands are connected to the brain via different kind of cellular structures. The idea that, and this is why it was very radical, that he was talking about, that there is a connection, like a physical connection through the cells, which has these synapses, and the synapses are flowing the information. For example, anything you do, anything the way you act, maybe your reflex action also, these are ultimately coming from this excitation inhibition of your muscle fibers going on to the neuron and all that. Right, so this is very important in terms of the theories of information processing. Now, little bit fast forward, and around the 1950s, right around the end of the Second World War, we had this experiment by Hubel and Wiesel, who actually in the right-hand corner here, they were looking now in the cortex, in the animal cortex, this is in the cat, and what they looked at, these are in the anesthetized cat, when the anesthetized cat was actually forced to make the eyes open and showing these kind of orientation profiles of different light bars. You can see that a particular orientation like this, let's say this is at 45 degrees from the baseline here, this orientation, there is a maximum activity, or what we call them is the firing of a neuron. These are actually something what we call is this action potential. You can imagine these are like binary zeros and ones then. So if there is an action potential, there is a line here, and these bars represent this action potential. So if something is tuned at a particular angle, there is more activity in a particular cell. And this is a very important sort of understanding in terms of if you want to understand information processing, because now different cells can be aligned, and each cell, one cell can be maximally tuned to this direction, the other cell can be maximally tuned to that direction, and so on and so forth. Now, if you kind of imagine the entire cortical area in the patch of the cortex, these all areas will have different representations or slightly different tuning patterns. Not all areas, each neuron in a particular area. Okay, so if you plot down this stimulus orientation in terms of degrees, and to the function of the neural response, which is the rate of firing of action potentials that is happening, you can see there is a peak at 0, which means this is the peak of the peak angle which this neuron is particularly firing. And then you can do the same curve for different neurons and kind of understand about all the cells what they are doing. Now, you have to imagine around the same time, end of Second World War and the whole period between First and Second World War, there was enormous development in the computer science sort of field. So where the ideas of von Neumann, Alan Turing was being developed, in the sense people were asking the question, how do you represent a code, or what is the definition of a code? So then this language leaked to the neuroscientists, so to speak. And what we know is the way we understand this now, we call this as some kind of a neural code. So for example, if you understand there is some kind of representation of the outside world in your neuron, you can call it a code. These codes can become complex. For example, a few years back, the Nobel Prize was won for spatial navigation, where people have talked about, like, if you release an animal like this is a rat, released in a sort of a square environment, and based on where the animal is at the time, so this particular neuron, there is a particular neuron in a specific structure called hippocampus in our brain, where that neuron will fire exactly when you are in a particular position of the room. So in a way, there is some kind of this is kind of an internal GPS in your brain, which kind of tracks down every locations you would traverse in a particular given geometry. Right, so people have talked about this. This is being called the place cell, and they have now talked about not only this cell, certain cells which are also called grid cells. They are not firing for only one location, but several locations. The point of making this slide is to show you that the codes can become very complex, and we can have complicated codes for even complicated way. So for example, how do you feel about my talk here today can also be represented by a specific set of firing in certain neurons. And that's what we are talking about neural code in a philosophical way. Now, more recently, and this is one of the ideas that has been proposed, the code obviously refers, I mean, it's itself, though it owes its birth to sort of, computer science literature, but then the code is some kind of a, detriment to the further evolution of the field, because code essentially means that you have code in a machine means that the code is fixed. But unfortunately, in the brain, this code is very much plastic. In a sense, it is changing with context. It is changing with learning. It can be changed with like real world environmental facts. I mean, events that you are encountering every day. So in a way, the coding may be not the best way to look at the entire brain information processing. So the way that we are arguing right now is something beyond this kind of coding principles that we can use to understand the brain information processing. Okay, so, and this comes to this idea that I had been talking about earlier, that, with Charles Sherrington's ideas that how do information is integrated. So this aspect was missing when the segregationist theories were being proposed, where brain areas being understood as kind of segregated into different structures. But now, since we are going into the future, we would like to understand how the integration in the brain is happening across different areas. For example, you would see more demonstrations of this, that you would remember me as a kind of a composite object of my speech, my voice, your visual representation, me as a combined whole. You don't identify me as these separate split things, even though the computer tries hard to do this, but you will still remember me hopefully as a whole composite thing, and where this whole is computed in a way where this integration in the brain is happening, because each area, as we know, as we understand from these different years of research in the human neuroscience, are doing different things. So where is the integration happening? So for example, one hypothesis is that, these visual areas, for example, how do the integration can happen? So for example, if you have a visual stimuli, the visual stimuli takes a different path after it reaches the visual target. This V1 is an area where information from your eye goes to via the subcortical structures like thalamus. So you can imagine your eye is here, and the visual information goes to first V1 and then to this part of the brain. If there is some if you are coding for, or if this information is relevant for something which is like remembering a picture, for example, which requires more time, versus it can go to this top regions of the brain, which we call the dorsal region. This is the ventral region. So this can go above, and this is called this what path, or rather the where pathway, and this is the what pathway. So if here, what we are talking about, the brain area, this one is not really doing everything, but this entire stream of areas along this pathway is involved in the information processing. This is just an example for vision, but you can expand these kind of ideas to different sort of other mental faculties, right? So moving on, beyond this, so how do we, this integration, what is fine is that we do understand there is a need for this integration, but how this integration is actually effectuated in the nervous system is the critical question. So this is one experiment done in rats, and this experiment is quite interesting because what they did this was a paper in this journal Nature, which is a very important journal for scientists. So now what they were doing here is that they were looking at individual cells. They were getting recordings from inside the cells, which are actually represented here. And they were actually getting these field activities, which is like EEG-like activities, which are placing the electrode on the top of the surface of the cortex of the animal. Now, what they see is that these wave-like structures are represented, like the trough of these waves is actually represented to the more activity of these individual cells. And these are excitatory and inhibitory cells. So what you get from this picture is that when everything is aligned together, you get a robust activity at the surface of the brain, or maybe as we measure human EEG from outside the brain. But when this alignment of this activity breaks down, you don't get that much of a robust activity, these web-like patterns outside the sort of, far away from where the action is happening. So in a way, the nervous system organization is actually organized in a way to synchronize these activities across different levels to give you an activity at a superficial level, which you can then observe. So these are actually, these ideas were further propagated by Francesco Varela's work, Ulf Singer's work, and what they have shown with different, and these are like outcome of several, like, maybe 1,000 different brain studies by different groups. We can start getting to look at, if we look at the human brain, if you put different electrodes, you can now talk about synchronization of activity, which is the EEG activity across different regions, which can be meaningful of some kind of an information processing going on, or more sort of a little more technically speaking of the integrative processes that the brain is involved in. So taking this idea of integration further, we can actually talk about in terms of this comes from these ideas of physics, from self-organization, from nonlinear dynamical systems, that you can use the concepts of synchronization to understand the coordination of activity of large groups of assembly, or the collective behavior of this system using non-equilibrium thermodynamics. And you can study the coordination instead of synchronization of specific cells. You can talk about coordinations of brain areas across different brain areas, or even coordination, so to speak, across different effectors of behavioral functions, such as hand been moving together like this. So this can all be kind of a coordinated action, and this can be reflected in the coordinated activities of the brain. So I will not go in deeply in each of these theories, but I will, because I have to cover some of the aspects that we are also doing in my work also. But there is another interesting sort of, from the perspective of computer science and the information theory, this idea of the idea of brain processing can be understood in terms of these Bayesian principles. So Bayes' theorem, as you know, in probability talks about you can sort of identify if you have a response and if you have a stimulus, which are essentially very stochastic variables. They can be sort of related with certain sort of conditional probabilities, like stimulus conditional on the response and response conditional on the stimulus. So stimulus conditional on response can be something you called a prior learning. So for example, if you think about this, that as we grow along, as we grow old, we are actually learning our associations with the stimulus. So we have stimulus, we have response, we have learned what each one of these response means vis-a-vis one stimulus. So our knowledge of the stimulus is conditional on this sort of response. And so we can form a good sort of idea of this whole stimulus space, so which can be also our priors, and we can predict, like given a new stimulus, you can now predict what are the underlying responses are, or vice versa. So if you have a response, our brain can also predict what is the stimulus there is. So why is this important? So imagine, so you have this is the eye, this is the retina. So retina is getting the images from outside, but what is it's translating into? It's some sequences of ones and zeros to the brain. And the way the brain understands it, it can't see the outside image, right? It can see these ones and zeros that we are seeing, that I showed you the earlier slide in Hubel and Wiesel slide, where you see these ones and zeros from this earlier level, and you have to somehow make or decode about what is there outside, right? So how do you decode? So you want to understand these ones and zeros related to your previous ones and zeros. Ones and zeros you have accumulated over your childhood, over your adulthood, and so to speak, like if you are an adult, right? So now the question is, these ones and zeros, if there is in a way, the brain is always also trying to predict what is happening. So if you think the brain's purpose in life is to predict so that you can survive your different environments, so then you have to do a matching between that prediction and what the things that ones and zeros are coming. And if there is a mismatch of this prediction, then you say, oh, this is an important thing. This is I need to remember. And this is what I need to process. If I can predict something, then anyway, it is not very relevant for me. So in a way, you can build up and go on and go forth until we sort of stop living. This process continues on. So this is the way a Bayesian sort of brain works, in a way that you have a set of sensory inputs, you have a prediction errors, and a mismatch of this prediction error will go into your memory, and you can then take it over for these other systems to remember. Now, so what I hope I have covered to all the sort of grand unified theories of brain function till now, and you can see a lot of them have been motivated by the language that have been used by computation and neuroscience. And this sort of slide encapsulates what a challenge, or what are the different kind of challenges that a multisensor or a multiscale brain has, and that needs to be bridged. For example, if you want to talk about a grand unified theory. So for example, you have different levels. So for example, if you look at this, you have different structural levels and you have different functional levels. So the structural level starts from proteins to chromosomes, synapses, cells, microcircuits, brain regions, whole brain, and to the behavior. And these time scales of these are like development and aging spans over different, like in years of aging life, whereas these protein scales are these vesicularly happening in the nanoscale, and you have to have devices imaging each scale. So now you can think about, we study this kind of behavior inside the scanner for different kinds of individuals. We can talk about learning, we can design, and we can also image these using different kinds of sophisticated microscopes, live cell imaging, and things like that. So ultimately, though, we have to bridge these different scales, and that is one of the challenge, I think, for any new grand unified theory of brain function. And this is something that has not been resolved yet. So coming to that, I hope I have given you a brief overview what a grand unified theory of brain function look like. So in a nutshell, all scientists till date in neuroscience are like secretly looking at least they're interested in this grand unified theory of brain function, because I think without that, people like Pradeep, who is interested in neuroscience, which he told about, they would not be interested, right? So this is an important part that it's a fun part, and that's also an important part that we really need to focus on. And it's also very useful in a way. I will come to the different when I come to show you some applications. So what do we learn from multisensory perception? First of all, what is multisensory perception? So for example, if you talk about different sort of vision and audition, so vision, as I briefly told you also, the light falls on the retina, and retina, it goes to the visual image, is formed in the visual cortex. And then you have this audition, where you have the sound falling on these ears, where there are structures like cochlea, which does some kind of a Fourier transform actually on the signals which are coming in, and they can send these signals, again, these codes, the neural codes, to the auditory cortex to like shape up your auditory perception. So across this, you have signals, you have percepts, and what you have developed is you develop concepts, right? So these, what we think is going on is in the brain is at the level of the concepts. So conflicts in this perception can actually provide us the gateway to understand the brain processing. For example, on the left, this is a sort of a static stimuli, first of all, right? So these are actually white dots, which are interspersed with black dots. But when you look at it, it gives you some kind of a dynamic sort of an illusion. And similar kind of illusion can be, if you hear this sound, I hope it plays. So what you heard is I think towards the end of when I played the sound, you were probably hearing some people, if you are using your speakers, you might be hearing nowhere, nowhere, nowhere. But this sound is actually just random noise, which are nothing really, or rather, so our brain tricks us into believing there is some meaning in this sound. So this is essentially an illusion. So there is conflict between the percept and concept when these illusions arise, and this is a way to study the brain in terms of the brain activities. So moving on to this multisensory perception. So for example, it is relevant to understand speech, because speech is a very much a multisensory phenomena. And this is different systems. I mean, different networks like auditory networks and visual networks interact when we are talking about speech perception. And it is definitely something, the most fundamental form of communication before Facebook or Twitter. Now, I would like you to hear this while watching this video closely, maybe towards the lips of if you focus on the lips of the vocalizer. And I hope you all have your headphones on. If you don't have your headphones on, maybe you can do it immediately if you have right in front of you. So I will play this now.
Pa. Pa.
So I hope that a lot of you probably heard tha, right? So when you look at the image or the vocalizer, now I will ask you to close your eyes and hear the same thing again. I will play it again.
Pa.
I'll play it again. Close your eyes. So if you close your eyes, you are probably hearing fa. So in a way that what we have done here, we have superimposed the lip movement of ka on the sound of pa, which gives you a illusion of tha. Now, again, this is a multisensory illusion. It happens when you are actually looking at the lip movement of the vocalizer and also hearing them. So the question we asked is, how do different auditory and visual systems are interacting with each other when you are having these illusions? This is an fMRI experiment across different age range, and we had total 55 volunteers. And this was a study done by my first student, Abhishek Mukherjee. And what we observed in this red line here, the subjects all reported what they are hearing during appropriate button press boxes. And this is the red line tracks the response of tha. This blue line tracks the response of pa. So if you see when the auditory and the video sort of the stimuli or the streams of this information are totally in sync together at zero lag, there is an increase in this perception, the illusory perception of tha, which decreases as we increase or decrease this auditory AV lag, which is the auditory visual lag. Meaning in this case, the auditory precedes the visual stimulus. Here, the visual stimulus precedes the auditory stimulus. And we look at these brain areas, and we find that this is the auditory area here. We call these the Heschel's gyrus, STG. These are areas included here which are active are Heschel's and STG. So across all these different lags, we see activity in the auditory cortex also is somewhat unchanged. There is an auditory cortex activation. Visual cortex activation is also present across all these lags, which is here. So visual cortex is here. But interestingly, there is this area which were known to be as multisensory areas, meaning they are active only when the presentation of multisensory sounds, maybe visual, auditory, visual touch, auditory touch kind of signals. So neurons in these areas are active when both these sensory streams are present. So these areas seem to be more active. So the activity is directly correlated with the illusory activity here, which means that these so-called multisensory areas are not really sort of processing for the stimulus per se, but they are actually also doing something related to the perception of the stimulus. And we actually proved this, and I would not get into the details of this. But again, this PCSTS, IFG, these are the important areas that are involved in this there. But so this is going by, the concept of segregation. And for the time being, you just need to know that segregated brain areas, which are actually physiologically different or separated, they are actually active, which we found, or they are more active during the task when there is a particular illusion. This is the long story short, right? Now, what we did next to understand the integrative processes, we looked at the Pearson correlation. Now, Pearson correlation, you can compute between any two time series. It gives you a measure. It's a mathematical measure of covariation of signals across two time series. Now, if I put a seed, which is in this red spot here, and I put this correlation of this seed's correlation with the entire brain, and I find out the statistically significant correlation, these are the areas which seems to be connected. So all these areas are co-varying with the activity of this area. And if you see, this network decreases, the size of the network decreases as you go towards zero lag, which is also close to this where the illusion was happening. So at where the stroke of the illusion, it seems there is the network that was present in the brain is currently suddenly disappearing in a way quickly, and there is a more activity in a specific set of brain areas. So the integration and segregation both are operational at the same time. So this we finally mathematically quantify by taking all these average correlations of the seed correlations from the STS across the entire brain. So as you see, these are blue lines represent the correlations done in the left hemisphere. There are two hemispheres in the brain, right? And then the red lines actually are the average correlations across right hemisphere. For both hemispheres, the correlation is decreasing, the brain correlation, along with the lag. This is the behavior. So this is a figure that is drawn from the behavior. This is a figure that is coming out of the brain dynamics, and you see how nicely anti-correlated they are. So when there is an activity in this area PSTS which is increasing, it seems the PSTS is actually desynced or desynchronized from the rest of the brain. So in a way, this decorrelation or desynchronization. So I was talking earlier in the beginning about the synchronization of different brain areas are relevant from understanding brain function. It seems the decorrelation or desynchronization is something also as important, because you can think about we have to desync from what we are doing to do the next thing. So desynchronization is something which defines us as human beings or attentive human beings, right? So we repeated this experiment using EEG. Why? Because fMRI has bad temporal resolution in the order of seconds, but our brain dynamics unfolds in the order of milliseconds. So this is done by my first graduate student, actually PhD student. He finished with a PhD in my lab last year, G. Vinod Kumar, and he's currently in Penn State doing his postdoctoral work. So he did this very interesting study where he looked at EEG dynamics of these different perceivers and the rare perceivers. So he divided the frequent perceivers are the ones who perceived. So even from the audience, I believe there would be a lot of people who are not perceiving the illusion, and he called them the rare perceivers. And he looked at their behavior, and you see there in the case of the rare perceivers, they are mostly hearing that fa, whereas the frequent perceivers, they were hearing more number of times the ta. And what he looked at, these brain dynamics unfolds like this somewhat in time. And you can talk about a measure called coherence, where you look at the spectral sort of determinants of this temporal coherence like we had done in the fMRI, but you can do very precisely now in the EEG. Once we look into that, ignore this part. These equations are how you compute these coherences and global coherences. I will, for the interest of time, I will ignore that. And what I will say is that this is a coherence map which is unfolding in time. This map represent any kind of red activity means the brain is synced at a particular frequency which is on the y-axis. Like this, look at this plot, the bottom right hand corner, right? So you have a map where you talk about the frequencies on the y-axis. This is time. These dots are the time where the stimulus actually appearing, the video. And there is an increase in coherence in this gamma band, which is between 30 to 40 hertz, whereas there is a decrease in coherence in the alpha band. So it means that the network which are communicating in alpha frequencies, in EEG we talk about these rhythms. Alpha is a frequency band which is the frequencies of which are around 10 hertz. Gamma is around 40 hertz. So gamma coherence is increasing and alpha coherence is decreasing. So when he actually looked at this individually across each participants and tracked their individual variability of perception, he found out quite interestingly that, say for example, somebody who had a very high alpha coherence, now we are doing it single subject by subject, right? And this is something that I want to come back, and Pradeep told me that I should mention this at least once in my talk, is actually this, I think the theme is talking about mastery, right? So mastery, what when you say the word mastery, the thing that sort of ignites in your brain is individuality of right? So everybody is not a master. Somebody is good at certain things. Everybody is good at certain specific things, right? So you master that thing. So in a way, illusion you can think about. Somebody perceives the illusion. Somebody don't perceive the illusion. So in a way, we are all unique individuals. And what you are seeing, that the way the illusion, at least from the mastery of the illusion is concerned, is happening, that the people who are perceiving the most amount of illusion have least amount of alpha coherence at the basal level. So when that alpha coherence can be easily broken by the stimulus, they automatically perceive the illusion. And this is what we have recently published in European Journal of Neuroscience. And from a sort of, coming down, I don't know how much time I have. I don't think I have a lot of time, but so I will skip the main maths part of this. But what we actually did this in the sensor space, which is in the EEG sensors. And then we can actually apply some kind of modeling tool and predict what is actually compute. Like you can simulate individual neural activity and simulate the whole brain area's activity and predict how this kind of coherence can emerge from the brain. And our lab actually, and in collaboration with my colleague, Dr. Dipanjan Roy, we are vigorously with several of our PhD students and postdocs are actually exploring these ideas that how can you create this kind of whole scale brain models, which you can then use to study synchronization and desynchronization phenomenon. And this is what we call the neural mass model. We can apply this neural mass model and predict what the dynamics is there going on in the source space. And now the validation comes from other tools. Validation comes from direct tools that can be applied from the engineering sort of physics, from understanding the sources. Like this is a detector. This is a classic problem of electrodynamics where you have infinite number of sources maybe, and there are a finite number of detectors. So how do you go to this ill-posed problem of finding out the sources? So turns out that you can use smartly the different knowledge of this brain folding and all that to actually kind of do a good guesstimate of where the activity is at the source level. And that source level coherences will then give you some validation of your prediction of the model. and I would skip that in the time being. But here I want to tell you about the main gist, what is the interesting part that, or knowledge that came out of this model, is that earlier I showed you that the frequent perceivers are having lower alpha coherence, the rare perceivers are having more alpha coherence, and we predict that this actually directly relates to direct auditory visual sort of connectivity. So it turns out that the auditory to visual cortex can be connected directly functionally, or it can be connected via the multisensory areas. So the multisensory areas like the PSTS. So what we are saying for the rare perceiver, this connectivity is very strong, whereas in the case of frequent perceivers, this connectivity is overcome by this auditory to multisensory and multisensory to visual connectivity. And exactly that we can see, at least for the direct auditory to visual connectivity, is more in the rare perceivers, which we see in these two plots here, that we actually do from the source localization techniques, which gives you a validation of the prediction of our model. So in summary, that what I can say is neural states are defined by states of synchronous oscillations in distributed brain regions. In a series of experiments in speech, with speech perception and visual attention, we have shown that decoherence of brain rhythms is critical when cognitive load increases or integration demands rapidly become complex, right? So decorrelation or decoherence is something that the future studies should understand, and the synchronization and desynchronization maybe can hold a key to understand or formulate a new kind of brain grand unified theory of brain function, so to speak. So what is sort of also important to look at here, these whole brain models give us a good windows, and this is where all these data analysis platforms becomes very relevant, that we can actually use this kind of data analysis platforms to go for using the whole brain models from structure to function, and actually talk about these important questions that are being faced by neuroscientists. And one of these ideas that we can actually take is we can go into the multiscale behavior, such as I have not talked about neurotransmitters like GABA, glutamate, which you might have heard in the context of mental health, sort of Alzheimer's disease. These different neurotransmitters or chemicals that control our mood swings, our behavior, our cognitive flexibility. These can be also tied up in the future model. So one of these things is actually that we have proposed is currently in bioRxiv and being reviewed in a journal. So hopefully that will come out soon, and then we can talk about more about these models. So I'd like to thank my lab, who actually have done most of the work that I have presented. So particularly my colleague, Dr. Dipanjan Roy, who is also a PI in the lab. And the work that I have shown you today was done by my first PhD student, Vinod Kumar, and also Shrey Datta, who was involved in a lot of this modeling work. Thank you very much. And I would be happy to take questions. Over to Pradeep.
Awesome. Thanks a lot for this wonderful presentation. I got to know a lot about what's happening in this specific area and where to go from here. One question I have right now to ask is, what do you see happening in next 10 years? What is this bold vision that you see that in next 10 years, this might happen, and these kind of use cases will spurn out because of the development in this area?
Right. So I think in the next few years, this will happen. So you see the list of this. These are actually big data repositories for data science aficionados. So Australia has this brain dialogue. I have listed some of the, in Europe, there is this, I think you can see still my screen, right? You can see my screen, Pradeep,
yes,
right?
Yes.
Okay, so Europe, you have the Blue Brain project, you have the UK Biobank in Cambridge in United States, so you have the Human Connectome project, the Brain Initiative. So you have all these different databases. And so the question is that we have only, these databases have fantastic data. I missed the animal databases no, I have not missed. So this Allen Institute has this beautiful mouse connectome. They also have, right now we have mapped the entire C. elegans networks, and these are also very important. So because we can understand a lot of, to tweak certain genetic sequences, and we can merge this with these genetic sequences consequence? How do they change the neural function? So these kind of questions earlier, which were not possible to ask without collecting more precise data yourself, can be asked now because we have this database, and that's where the field is moving. I think we have a lot of very interesting data sets that are being developed, but we need sort of, smart people actually to handle these data set to come out with good insights of what is really the thing that is happening. And there lies a lot of answers actually. If you look at, even if you take into current COVID work, if you see a series of work has been involved using application of AI in discovering maybe some repurposed drug that is already available can be used for COVID and treatment of patients. So those kind of analyses are actually handy here as well. Like if you have a particular mental disorder, can it be linked to a specific gene? Then can it be linked to a specific, once you have a gene, you can have a specific drug target. Maybe if you have a particular molecule you can link to, you can start talking about better drugs. Everything can be just better if you can handle this data very judiciously and effectively.
Got it. Thanks for sharing the answer. So we have some appreciation coming in from the live stream, people who are watching at live stream. And there is one question I'll ask on his behalf. How scientists had determined brain's areas such as amygdala? Did they use trial and error method?
That's well, I mean, in a way, sometimes these areas are serendipitous. Like a lot of this, like let me go to that slide. Anyway, so if you let me slowly go down while I speak, if you are okay with that. So there was this slide where I talked about this neurologist, neurosurgeon like Pierre Broca and Wilder Penfield. So they actually, when they were neurologists, and these things come to in a clinic every day, like people are injured, people get a lesion in a particular region of the brain, and they lose certain functions. So the speech area, Broca learned about this area from a particular guy who lost ability to speak. And what he found out after the guy died is that there was a lesion in that particular area. That's how he came to know about this, that area may be relevant for speech. So this is how this whole aphasia has been. And there are beautiful books written by Antonio Damasio on this, who is also a neurologist. We have this famous patient, HM, we have who actually, to treat his epilepsy, he had his entire hippocampus removed. And he kind of, then went on, and what people found out that he couldn't form new memories. And, this idea that entire region removal can actually remove memories. You have seen it in some movies, famous movies also. But that idea came from this particular patient, like who had his entire hippocampus removed, but he couldn't form new memories, but he could perfectly remember his old memories and do everything else in life, like eating, drinking, all this stuff he could do. But it's just that he couldn't form new memories. So all these, so a lot of these case studies have been useful for a lot of these. And I bet you that amygdala may be one of these case studies, that maybe somebody once had a lesion and then you had a problem with actually emotional well-being or fear psychosis. And then, of course, there is this famous works by Joe Ledoux and his group in NYU who have furthered our idea about amygdala, how it is related to fear processing and emotional processing in general.
Got it. Awesome. Thanks a lot for the answer. So this presentation, there is a lot of information over there, and much useful information. And probably if I go through all of that, it might take years or months at least to know more about it. So as a beginner, what would you suggest that this is where you should start, or these are few things, top 3 things, where you can get started to know more about this field and probably build something out of it, something useful?
Right. So, so there are these very nice books written. So there is one written recently, I mean, not very recently now, I think 2010, 12, by Yuri Budzaki. He's called The Rhythms of the Brain. If you want to understand what are the different signal processing perspective of the brain rhythms, what they mean and all that. And of course, if you are interested in sort of, get a snapshot of how people came up with these ideas of the brain, of what each areas do, you can look into Antonio Damasio's book. Descartes' Error is one good book, and there are others. There are Oliver Sacks books also that, of course, different books he has written over the years, and there's famous movies like Awakenings. So all these books are actually very relevant, the popular science, for an incoming student to ignite their interest in the brain. And I would say that, you have enormous amount of information handy right now, Google, and, you have lots of journals, which is a very exciting thing that is happening all across the scientific community, are coming open access. So most journals are actually, you can freely download the papers. And, right now, earlier, like, you would have boundaries that you cannot read from a certain area of the world, that you cannot have access to papers that are not that you are not bounded by that anymore. You can just download most journals. Most good journals paper can be downloaded right now using these open access ideas. And if you, the other option is you just send the authors an email, contact them by email. They will be happy to sort of share. And, neuroscientists, we love to talk about the brain. We love to philosophize about the grand unified theories of the brain. And if you ask this kind of interesting questions, which are, scientific, I mean, I'm sure people will respond, and, that's the way to go.
Got it. Awesome. Thank you very much. So about this talk, I think these are all questions that we had, and I can ask more something that is not related to this. How scientific community is dealing with COVID-19? Are there any specific challenges specific to scientific community?
Yeah, of course. I mean, see, this the main challenge is that, developing, working with a live virus like this, because what gets omitted in this picture is when people are actually, some of my colleagues are working with it, this live virus, there is enormous risk, because after all, wearing the PPEs and everything, people who are actually doing the vaccine work, even forget vaccine, people who are doing testing work, they are at enormous risks. Right now, for example, I live in North India, close to Delhi, Gurgaon. So there was a hospital in Faridabad, which is in Haryana, close to, again, Delhi. So the entire hospital had to be shut down because 50% of the people, the medical people, were actually infected people, because there was testing going on. So if you have faulty kits and all that, so it's a problem, because this PPE is a huge issue, because the virus seems to be very contagious, and it just spreads very quickly. And that's one of the big challenge. How do you protect? We talk about protecting the frontline workers. How do you protect the scientists who are working with it? So that's a big challenge for the scientific community. And top with that, when it comes to vaccination, the way our right now clinical trials work, and that also takes its own course. That there are certain checks and balances which are also required for other reasons, and that's also a cause that is this vaccination is delayed in a way. So, but I hope that would come soon, as soon as possible, like everyone is in the world looking to it.
Awesome, great. So thanks a lot for this presentation. For the audience, you can go to live.gitcommit.show, and if you like this presentation, you can hit the clap there, and also do share with us your feedback about the talk, about how we are doing, and you will have about 30 seconds starting from now to clap. So just let me. Okay. And Arpan, would you like to share any last message for the audience? And that will be the closer of this session.
I think, I don't know who the audience are, so I can't see them, but I hope there are a lot of young people, and, they are very much into neuroscience, and I hope they join neuroscience and, take this as a career option, and, and do the grand breakthroughs which are possible now that we have the data. And I hope at least I could motivate some of our younger scientists in the making, kind of doing this work. And of course, there are different places in the world you can go to, but I think that scientific temper, and for other people who don't want to do a PhD and all that, or who don't want to join science, I would say that, we should build a scientific temper, not only in this country, but across the entire world, and, trust the scientists, and in a way that would be helpful for the entire community and the entire society.
Great. Thank you very much, Arpan, for joining in and sharing this knowledge. This weekend is going very wonderful right now. A lot of things to learn.
Thank you, Pradeep. Thank you for organizing this. It's a great sort of, I would say, effort. I think it requires, I have organized conferences myself, and Trust me, the physical ones are difficult, and I'm sure these virtual ones are equally difficult. It is not easy. So you guys have done a commendable job sitting at your homes and actually organizing them, and I'm sure that probably you also do some other things outside and to get this spirit going is great to see. And thank you to all of you, whoever it is part of this Invide Lab that's also work. You guys rock.
Awesome. Thanks a lot. Thank you for those kind words.